Okay, I think we can make a start. Welcome everybody to the BioPhorum Drug Substance showcases. This is our final session for the year. These are quarterly sessions that we've run through 2024 to introduce our different work streams that we have in the drug substance program. So if we take a little look at our agenda for today, it's a very short introduction from me, and then I will be passing over to Olivia and the raw materials team for a much more interesting overview on the raw materials workstream. And then Olivia is going to be passing the baton over to our colleague Sean Collins, who is going to give a bit of an introduction to the lifecycle management workstream and also to the continued process verification community of practice. So if we just start then with a short introduction. So welcome everyone on the call. If you are familiar with BioPhorum, you might be familiar with drug substance already, but we know that today, so this session is very much open to all. And if you haven't been part of BioPhorum previously, and this is new to you, welcome to what is quite a unique collaboration and trusted environment for the biopharmaceutical industry. We are in BioPhorum organised into different forums, and the team speaking to you today is representing the Drug Substance Forum. My name is Nicola Powell, and just to tell you a little bit more about drug substance, you can see on the slide here, we are very much an operational focused forum where we bring together drug substance subject matter experts and leaders from the major biopharma manufacturers all over the globe and our major supply partners, and work on some of the main key challenges that matter to us as an industry. We hope to inspire adoption of new technologies. We absolutely expect to shape the regulatory landscape and be able to provide publications and tools that help us to adopt our operations to new guidance and apply risk-based approach. We look at increasing process efficiency, improved asset utilization, and most of all, confidence in retaining licenses for our members. So these sessions we have set up really just to provide a very high level introduction to the different work streams in our program. The idea is this is an informal session, so please do raise your hand or pop any questions you have in the chat. You should find that your microphones are all enabled if you keep those on mute during the presentation, but we do invite you to ask questions and come off mute and interact if you wish to do so. And we will make the slides and the recording available after the call today if any of your colleagues might be interested and weren't able to make it along. So the second part of the introduction from me is just to show you quickly the drug substance program, what our current work streams look like. So you can see listed across the top line here the different work streams we have in drug substance, and listed below the work streams any sub teams that are active for those particular work streams. So highlighted in orange, we have the three areas that we are featuring today. So without further ado, I will hand over to Olivia, who is going to introduce the raw materials program. Thank you, Olivia.
Thank you, Nicola. Good morning, everybody. Good afternoon. As Nicola said, my name is Olivia Paisley, and I'm one of the global change facilitators here at BioPhorum, running the raw materials program. And today I am joined by also one of our members, Hesam Mahjoubi from Amgen, who will also be speaking to you on some of the work that we are conducting here within the raw materials programme today. So I'll start with a little bit of history of the raw materials programme, and then we'll move on to what we are currently doing. So next, there we go. So the raw materials programme started back in 2013, and it started as a work stream focusing on raw material variability. And it was the first work stream to combine both the expertise of our biomanufacturers and our suppliers together here at BioPhorum. We had several key publications starting from 2015 onwards, including one looking at patient-centric requirements for the supply of raw materials into the biopharmaceutical manufacturing industry, and a raw materials risk assessment guide, which was developed through industry collaboration. It was after this and some other further publications that it was decided by senior leaders within drug substance that we should be promoting this to a programme of work. And in 2021, the members set out to develop a strategic framework to underpin the programme. Common to all programme members was a commitment to solving technical problems relating to the harmonisation and management of raw materials in the biopharmaceutical industry, and the vision to control the raw material supply chain, manufacture on time, and deliver consistent quality to our patients. That raw material strategic framework is built on 4 pillars that drive towards the stated vision and mission. And these are 1, to advance our industry knowledge, 2, make second sourcing a reality, 3, enable predictable raw material performance, and 4, optimize limited lab resources. They're underpinned by the regulatory and quality environment that the industry operates within. And again, the strategic framework cemented the important interaction and cooperation of our biomanufacturers and suppliers to achieve the vision and mission. Our raw materials programme is set up and combines a strategic work stream and several technical work streams. Our strategic work stream combines our joint leadership team, and they are an empowered leadership team that are working currently to define a 5-year strategy with actionable and time-bound deliverables for the programme. They also provide technical guidance to our technical work streams and have a sort of start, stop, continue governance over them. They also receive updates from the technical work streams on a routine basis and can help with any issues that the technical work streams encounter. And we also host other work streams within the wider BioPhorum network and provide a platform to them to come and present on topics where raw materials may touch. They also receive the majority of our Ask BioPhorum questions into the joint leadership team due to the wealth of experience that we have within our JLT members. Currently, we have 1 current technical work stream open, which is focused on media and analytics, and this has been actively publishing since 2020. And currently they're working on a tool and guidance paper to identify critical material attributes for key raw materials for cell culture. And we'll talk about this a little bit more further into the presentation. We also currently have a space open for a new technical work stream, and the work that the JLT are completing right now is to undertake a task to identify a suitable new project or topic for that area. We have had several topics proposed and discussed within the last few months, including discussions on compendial and non-compendial filing strategies for raw materials and looking at ways to simplify the clinical to commercial tech transfer process in relation to raw materials. However, the topics that have currently been voted the highest so far by our JLT members are around the simplification of the inbound supply chain, specifically in relation to questions around what are the minimum standards for qualification of different raw materials, and can this lead to a reduced sampling and testing strategy for the industry? And if yes, what does this look like? And can we look to develop a guidance to support the industry on alignment? If this is something that you might be interested in, we will be looking to further scope out this simplification of the inbound supply chain large topic further at the start of the new year. and we will be looking to recruit SMEs that have expertise in this area. So please, if you are interested, do get in touch with myself to find out more. We've published a number of publications, guidance documents, tools, and benchmarking surveys since the Raw Materials Programme initiated, and several of them are shown up on screen here. However, we have published some key publications. So one around a raw materials risk assessment, and this included a shared language between our biomanufacturers and our suppliers. Two documents on a BioPhorum approach to the registration of innovative raw materials using quality by design principles. The first paper was looking at using virus retentive filters as an example and applying the QBD principles to this. And then we published a second appendix, which was then applying this to protein A resins. And finally, we have published a paper discussing the source, impact, and variation of trace metals within cell culture media. And it's work from this publication that's been taken one step further by the work of the media and analytics team with this guidance on critical raw materials in cell culture media. So with that, I would like to introduce Hesam, who is a senior process development scientist from Amgen, and he's one of our key contributors to the work of the CMA team. Hesam will now talk you through the aims of this team and the deliverable that we are trying to create for the industry. Over to you, Hesam.
Thanks, Olivia. Good morning and good afternoon, everyone. So today I'm here on behalf of many folks that has been contributing to this work stream in the past 2 years that I have been involved, and it's a very exciting, very complicated project to work on with a lot of up and down. So I will just try to give you a brief overview of what's the intention and where we are standing on. So next slide, please. So our goal in this work stream is to understand and define the critical attributes for cell culture media with a very, very deep dive into all the subcomponent of the media that is being developed or in the process already. So what are we trying to achieve in this work stream is, first of all, is to bridge the gap that exists in the terms of that how you are able to systematically have an approach to really identify what are the actual critical attributes from all of this N-1 material that comes in and in the final form of the media can impact your process. Furthermore, we are trying to enhance the alignment and understanding between the suppliers and manufacturers of the media and biomanufacturers in the terms of the criteria that we need to agree on for testing and control of all these raw materials that go into the media to make sure we have a good control on the final media that we are going to use in manufacturing. Also, we're going to try to have a real life example and very specific instances of how, for example, individual subcomponents based on the experience of suppliers and biomanufacturers had impacts on the final performance or product, and then how it can improve the design of the media during the design stage. And of course, in the end, as I mentioned, we want to make sure that we get all this alignment to just put everything that is critical for testing and not just put some default stuff that we see now in many cases that is already put on the media specification that are not necessarily that value added, to just make sure that everyone is on alignment. Next, please. So how we are going to get there? First of all, to get there, we are trying to build a guideline and a template, which that template will funnel into a tool in the end that will leverage the QBD principles for a structured framework that you can build to get to what are the actual critical material attributes that I need for my media. What we are gathering is that we are collaborating with a lot of suppliers and biomanufacturers and their networks to just survey and get as much as information that we can get from the experiences that they had with the issues, with the subcomponents during the manufacturing of the media, to funnel it into this framework that we are building. And the goal, the ultimate goal in the end is that based on all of this information, we're gonna publish a white paper and a guidance on how you are able to use this QBD-based tool, the templates that we have, that if you have not much experience or you're a very experienced person and you're just looking for a specific details or issues that you think might be important, you can just quickly jump to that piece and then figure out that if this is a critical attribute or not, or you need further control on the subcomponent or not. Next slide, please. So how are we going to get there? As I mentioned, based on the QBD design, I'm not going to go through the detail of it, but there are 5 elements that we are looking into building that template. Is definitely the intended use that could have a lot of things like the concentration, the target cell lines, the buffering, the pH, the osmo, the problems with the process compatibility, like if there are impurities that you have in there, if there is any impact of bioburden or endotoxin within the media powder that you are storing in the warehouse till you get to the using of it, problems with the trace elements, residual solvents, and anything else that could marginally, in some cases, that you might not pay attention to it, but in the end, after a while, people have seen issues with the stability and storage, safety, and I mean general quality requirements, the assessments for adulterations, nitrosamine, melamine, and other things that could be important that you might need to consider if you want to put more controls on the N-1 as well, or have at least those conversations with your supplier to see if everything is robustly controlled. Next, please. And based on that, we have categorized this QTB into 8 cell culture media components that each of them we individually are looking into what is needed, what is expected to have. And these are just the typical things that you find in many of the medias, the antiphons to organic salts, inorganic salts, amino acids, sugars, vitamins, and etc. Next, please. So to achieve this goal, definitely one of the important things is the collaboration and transparency, and of course the knowledge sharing between the biomanufacturers and media suppliers to come in, share their experiences, the issues that they had, the investigations, for example, they have done on certain components or issues that they had and what they found out at a high level. So we can just put all of these together as those real life examples and just mentioned. And then this will help to build that QDB template, as I mentioned. Next slide, please. So, overall, these QDB templates will help us to define a control strategy for our media critical material attribute definitions, and it just boils down to several control elements that we are considering when we are building this, which includes the formulation design as a part of those evaluation of the N-1, those controls that the supplier has on those N-1 materials, the type of the technical assessment and risk assessment that will be done in case that, for example, the testing is not still there in the terms of testing. Of course, the qualification, the stability studies, and then so on, that is already, I know that plenty of suppliers is already very established, but we're going to review all of this. And then, of course, the technical assessment is just a foundation to all of these to justify and derive many of these things. Next slide, please. So this is just a brief example of the initial matrix that we are building, that it will boil down to that QDB template, and it will be part of that final publication and the tool. So as you can see, we are categorizing each of the subcomponents, like in this case, amino acids and vitamins that you see. We define in that template that when you're looking at it, what are the ideal requirements that you want to have from both the biomanufacturer side, from the supplier side. We identify the impact area, then we are in the process of gathering all the information as much as we can from the suppliers, biomanufacturers, and examples, and then how those examples boil down to the impacts, and then what are the control strategies that they have. And then we go with the general what's the level of the impact, the variability and detection, and then when this is completely filled, it will be in some cases broken down to, for example, specific amino acids. We might go in more details for it, and then all of these will be hopefully funneled it into a form of an online web tool that, for example, you are able to put a keyword that I'm interested in X, Y, and Z, what you can give me, and then it will just put for you more information that if you're concerned about this, these are the examples, these are the impact area, and you might want to consider looking into X per prior experience that, for example, someone had in their process and the result of their investigation show that. So it's still in the work in the process, and you can imagine there are hundreds of N-1 components. So we are trying our best to go as much as we can in the details to make sure that we have a very comprehensive package of information that none really exists in the industry, and we are hoping to publish this probably in the next year if everything goes well. And
but just again to say, as Hesam said, we are doing a lot of work with the CMA team right now. So please, we're still in the early stage of gathering data. We will be looking to supplement that with input from many suppliers and biomanufacturers. So if you would like to get involved, again, please reach out. And again, on the point of our new technical work stream within the wider raw materials programme, again, we will be looking to recruit into that at the start of next year. So that is completely open for anyone that has an interest in that area to join us as well. There's no questions then. I will thank again Hesam for joining us and sharing that information. And I will pass over to my colleague Sean. Sean, I think we might do a quick switch through. Are you going to share your screen now?
I am, yes.
Perfect.
Give me a second, just for ease. Right. Hopefully you can all see this now.
Yes, we can.
Brilliant. Okay, well, first of all, hello all. Thank you very much for joining today. My name is Sean Collins, and I'm the Global Change Facilitator in charge of the Lifecycle Management Workstream, which I'm here to talk to you about. So lifecycle management is something that we all have to contend with on a regular basis, whether that be as a result of a conscious change to improve a process, accommodate increased demand, or satisfy new markets, or it could be an enforced change due to things such as material supply changes or regulatory guidance impact. Either way, it's something that takes a lot of time, resource, and energy to maintain. So as a result of this, the Drug Substance Forum members and leaders start talking about developing a work stream to try and find a way to reduce the burden of these changes. The discussions on this began in 2020, and it's quickly identified that lifecycle management of post-approval changes had many areas that could be improved by collaboration within BioPhorum. It was identified and agreed that they were a constant source of pain and work, that changes can take multiple years to fully implement and be approved in all global markets for a particular product. One member even cited that it can often take them 8 years to get approvals for one across all the markets in place, which is just outrageous, because by that point, you probably already put other changes in place to get follow up with. So it's not really sustainable. It's something needed to be done to try and streamline the process for a lot of these changes. It was then agreed by the group as well that there was no real standard industry approaches to a lot of common lifecycle management changes. And this is an area where work at BioPhorum, our members could outline some best practices with the hope of streamlining and improving speed of execution and approval of these common changes. The discussion initially identified a lot of problems with obtaining approvals could be improved with a well thought out submission package in the first place. That would help protect the product lifecycle and minimize the amount of time and costly post-approval submissions. So whilst that's not really part of the Drug Substance Forum's focus, a separate work stream is established in the RegX group within BioPhorum to address this, and the discussion returned to what we can be done as a drug substance processing perspective. So it was then agreed that a valuable area to explore was around validation approaches to common lifecycle management changes. So what you see here is the workstream charter. That was developed by the team, and this governs what the group's focus is. As you can see, it identifies the main problem being a lack of industry standards, which leads to variability in internal and regulatory expectations for requirements and validations for lifecycle management changes. This often then leads to a disproportionate amount of work and the data being required to be submitted to health authorities, which is not commensurate with the risk of many lifecycle management changes. The goal of the workstream is then to work together to share best practices and internal standards for common lifecycle management changes to create industry standards and best practices that will reduce the validation burden, as well as drive consistency as to the level of documentation needed to support those validation strategies. We plan to do this by creating various deliverables, such as white papers or guidance documents and tools, to address specific areas of concern as they are identified. Now, lifecycle management of post-approval changes is still a very vast subject topic, and so one of the key and first things the group had to do is to identify an area for deliverables to be created, and this is something that they have to continue doing as well. So the workstream was launched last year in 2023 and currently has one deliverable nearing completion and a second which has just begun, and the team are just starting the scoping exercise to really finish off the paper, so get the ideas ready for the paper, and then we're also starting out the next scoping exercise of what the next level should be, and I'll talk about all these different proposed deliverables in a moment. So I've explained the principles what the workstream was started for and what its goals are. However, I also wanted to share why our members feel this workstream is valuable and what they expect it to achieve. So these are some quotes from members. They believe the group will help harmonise expectations. This is from both the manufacturer but also regulator side. It'll allow our members to learn from each other's best practices and define industry standard approaches or definitions. These standardised approaches will then encourage the usage of scientifically justified data packages in order to changes to be as efficient as possible and to streamline and increase the speed of the lifecycle management changes. All of these points are at the core of what this workstream hopes to achieve. And the more members we have involved in this group, the wider range of opinions and best practices can be incorporated. So if any of you are interested in joining, we're always going to welcome more people to come get involved. Because this will allow us then to define and create globally applicable best practice guides for a multitude of common lifecycle management changes. So I mentioned we have 2 deliverables underway, and these are governed by almost a sub-team structure, with each team focusing on creating identified deliverables. We have the main workstream team at the top, which focuses on reviewing content from the papers as they're created, as well as identifying what's more important and what's next. So we've got this kind of continual process of reviewing, scoping, and then creating teams to address the different deliverables. The first deliverable was focusing on equipment changes and specifically looking at functionally equivalent equipment changes. The second, which is just beginning, is on expanding operating process parameters and how best to manage this. And then a third team, which will be established at some point in 2025, will form once a new topic has been identified and agreed. But this is likely to be something around materials or site transfers, just so we're kind of covering the different aspects of the types of changes we can incorporate. The functional equivalence paper had an agreed focus of trying to define functional equivalence, because it's realised there is no standard definition for this, and it's identified that member companies often classify this differently. They may interpret differently, they may use different language, but it's ultimately means that there's an awful lot of, it leads to a massive variation in what members file, which in turn makes health authorities question what's been provided to them, as it may not always meet the same standard. With the paper, we hope to provide a set of agreed minimum parameters that need to be assessed in order to prove functional equivalence of various pieces of biomanufacturing equipment. This will then support the desire of health authorities getting used to accepting a scientifically reduced data package on a more routine basis. In order to bolster this as well, we'll leverage case studies from our members to provide real-world examples of where the streamlined data packages have been successfully approved by health authorities to further bolster the message of the paper. And ultimately, what we want to try and do is get in a position where everyone can look at this and say, all right, this is the best way we can define functional equivalence to make things quicker and faster. Our next level, which is on the expanding operating process parameters. What we identified here is that, again, comes down to lack of guidance. The requirements for expanding parameter limits varies massively based on the criticality, and the guidance that exists there is very minimal, and we could do with having a set structure for this. As long as you go by this process, cover these parameters, then you should be able to expand this without any particular problems. It means that you can do things quicker, faster, less assessment up front, and just get on and do it. So the purpose of the paper, as you'll hear, is to create a guide on the most efficient way to do all these. So again, ideally starting at what should be the minimum requirements, scientifically justified, to expand different classifications of parameters, process parameters rather, and sorry, I lost my thought there. In order to define the class names, and then also provide essentially the guides for this. What we ideally like is that it should be a tool that can be then used to adapt to it. It's not going to be this is what you need for this parameter, this is this parameter, because that's going to be too big and clunky. What the purpose of this paper will be is to be a tool that is adaptable to multiple scenarios, providing that framework of what is required. And yet again, we want to try and do, because we think that this is incredibly valuable for members, provides evidence. We intend to leverage case studies to outline the best practices that members have used and had acceptance by health authorities. Because we do believe that including those real life examples is valuable in, I suppose, getting acceptance with the industry and market, and gets health authorities on board as well. And then just for simplicity's sake, we've added on the side that some of the expected benefits we see here. Again, if we're able to streamline these approaches, dictate this is what you should do for expanding this parameter, this is what you should do for expanding this parameter, then we'll see a reduction in the experimental data and timelines, streamlining of submissions to regulators, a consistent approach, reduced deviations due to having wider parameters, because we all know how easy it is to reduce and tighten parameters, but expanding ones we often don't like to do because we know it's going to be challenging. But if we can have this essentially guide out there for this is what you should do to scientifically justify it, it should be easier and less of a burden to do so. And if that's going to reduce your burden on deviations and the resources required to address all those, that's surely a positive. And finally, on this paper, we just want to kind of just focus, again, for ease, for you all to see, the focus of this that's identified is within that red square. So we're going to be looking at critical process parameters, key process parameters, and non-key process parameters based on this diagram. Each of the parameters will be assessed on what is required and different guidance provided for each. This will specifically be on process inputs, not outputs, and the raw materials equipment parameters are out of scope, because again, we've tried to break our teams into, we've got equipment, this one's process, and then we hopefully have another one in the future on raw materials or something else. So that's why, again, specifically this is around the process parameter, but it's just to try and demonstrate you, this is the kind of thing that we're looking at to try and benefit the industry. That's what the team is currently working on, but then it's what's next. We have got the lifecycle management. Sorry, the topic of lifecycle management covers a vast array of activities. Even when limited to validation activities, there's a lot of different areas of potential pain, a lot of different areas that can benefit from industry collaboration to define the best practices and standards. To that end, the team are continually looking at what areas should be considered next and assessing based on what would have the largest benefit to the industry. As the team, and as mentioned already, as the team have a sub-team focus on equipment and another on process, it's expected the team will either focus on something regarding materials or something to do with site location or batch size. So here are some ideas that have been suggested just so far as possible as areas to consider. This list is not exhaustive, and the team have worked hard to capture as many different avenues as possible. This is to show you some of the things that are being considered for future deliverables, and just a flavour of the work that the team have been looking at to kind of capture all the different problems that we can assess. I think we believe that this work stream, the lifecycle management work stream, has a great amount of potential to deliver some excellent works of collaboration with the industry, and over the coming years we expect to create multiple deliverables, all providing what we hope will become industry accepted standard definitions and practices for managing common lifecycle management changes. And this will then in turn allow all manufacturers to streamline their lifecycle management processes and ultimately get medicines to patients faster and cheaper. As that's ultimately the goal of what we at BioPhorum here stand for, and it's one I'm quite passionate about, really. So I'm quite keen to get behind this. And that is the lifecycle management workstream in a nutshell. So I would very much be keen for anyone to join this workstream if you are interested in learning more about it. If you are interested in learning anything about the topics that's being discussed, what can be coming next. If you want to get involved, please do reach out to contact me. But in the meantime, does anyone have any questions? No? We're going to assume not. In that case, then, we'll carry on. And you're still stuck with me for the next one. So, as I said, stuck with me. So now I'm going to be speaking to you about the continued process of education community of practice as our final showcase today. I'm also the facilitator for this group. However, as it is community of practice and not a work stream, this is not managed in the same way. Our communities of practice are chaired by our members, and right now we have 3 co-chairs. We have Scott Kincaid of Pfizer, Priya Bopana of AstraZeneca, and Henk van Urk of Thermo Fisher. The way that works is each member takes it in turns to chair the sessions, lead the discussions, and engage our members, and they're supported by myself to help deal with, say, the admin side of the actual running of the community of practice. So before we go any further into kind of what this group's about, how it is, I wanted to briefly talk about what a COP is or does. So a COP or community of practice in BioPhorum is a platform for like-minded and skilled members working in the same field to share best practices and emerging trends without the expectation of creating a deliverable or publication. That's the key parameter difference of this, is that a work stream comes together with the goal of creating something they're going to deliver and put out there that will ultimately try and provide benefit to the industry. A COP is formed when a work stream has completed all its objectives, but believes there's still significant value in continuing to interact. So this group, the CPV1, was formed a number of years ago. It's achieved an awful lot, which I'll come to shortly. But right now, when they finished all the work they'd done, they believed that it was critical to stay together and continue to interact, share knowledge, because the CPV space is it's a constantly emerging and evolving topic, and it's one that needs to. It's having that venue to interact with your like-minded peers is beneficial. So I mentioned briefly already, but a COP, the main point is the facilitator aided, but as I say, mainly self-governed. So the members, they will bring forth topics to discuss. They are allowed to present or put forward presentations from the organisations to share their best practices, to discuss new guidelines, to discuss new resources they've identified. It's very much down to our members to decide how they want the COP to run, to operate, what the focus should be. It's much less because it's more about how our members can interact, what they find most valuable from it. Yeah, sorry. So yeah, what I want to talk about here was the history of it as well. So this particular work stream started in 2013. I say over the course of those years that was together, it did publish 6 publications, and then after that, it converted into community of practice in 2020, and it will continue to be one for a while. For now, it will continue to be discussing what's needed by our members as and when required. If there ever was a desire by the members to dive deeper into a more topic and create something content, there'd then be the option to have a discussion with the leadership of BioPhorum, the Drug Substance leaders, to say, could we reform a work stream and create some content, create something again, because we believe there's value in this. But for the time being, as a community of practice, it lives on to share knowledge and hopefully just learn from each other. So this was a very active work stream, and with various teams set up over the years to create valuable publications in the space CPV. Like I say, they were also very active in sharing their best practices, benchmarking each other, sharing resources they'd made or provided, and then asking questions via the Ask BioPhorum feature, which if you're not aware about, I'll explain a bit more shortly. And all of this wealth of knowledge that they created all sits available for a member community. So whilst it's been around for a long time, it's not all gone. Everything that was shared, discussed, or created all sits on our shared platform known as the Hub. So if any of you were to become members, you'd have access to all the historical documentation going back to all 2013. So it is still even if you didn't take part in after discussions, there's a wealth of information there to get involved in. So as well as the other information shared and available on the Hub, these 6 publications were produced by the group over the years of it being a work stream. All but one of these is available as free to download the BioPhorum website. That other one is then still you can still follow a link to it from the BioPhorum website, and where you can try and download it from there. So if you are interested in any of these, simply search for these titles on the website. And as you can see, they have been downloaded numerous times, and so have helped a number of member companies in their processes with regards to CPV. So I mentioned it already, but as a community of practice, they no longer create content, but continues with shared learnings, best practices, and discussing new trends or guidance. Discussion topics are suggested by members and discussed during team calls and voted on by members for interest. And so at the moment, the group's just gone through a ranking exercise, and so these are the most highly ranked topics from the exercise they just completed, and this is what the group will be looking to discuss in its upcoming meetings. So you've got like obviously it's quite varied, but this is the tip of the iceberg of what they all wish to discuss. These are literally the ones our members believe are the most valuable. So those of you who are interested in CPV can see that if any of these are interested to you, these are going to be discussed in the next upcoming meetings in the new year. So please do get involved, and there are some very good opportunities for discussions there. And one of the other things I mentioned, Ask BioPhorums, this, for those of you who may not be aware, I'll mention it briefly. This is the tool that exists within BioPhorums Hub. It's a online Q&A tool that allows our members to pose questions to the rest of the group day or night, regardless where in the world you're based. And it means that you can interact and collaborate and learn from each other without having to wait for a meeting. A web meeting for the group. Because a community of practice, one of the other things of them is they meet less frequently. A work stream will typically meet once a month, say, depending on what the requirements are, maybe every 2 or 3 weeks or maybe 5 weeks, but a community of practice will typically meet every 8 weeks, 10 weeks, much less frequently. So it's less of a burden on the chairs and less of a burden on the members to take part, but that therefore means you've got less opportunity to interact. If every 8 weeks you've only got 1 hour to talk, you still want to be able to interact with us. So Ask BioPhorum is an absolute critical and vital tool for our communities of practice. And it provides a great opportunity to put simple questions out there or complicated questions, but that then often leads to bigger discussions. So these are just some of the recent topics, headlines that have been put forth through Ask BioPhorum by members that have been discussed for on the hub, which again, all those results and interactions remain there, so future members can come back and look at them. But then these were also taken to our web meetings and then discussed in more details as a prompt. So it's not a matter of we have these ranking exercises where we go through, say, these are the topics we want to discuss, but it doesn't stay like that. It's not always just stuck on this is what you must discuss. It's very much an open platform for our members to talk about what is important to them. So it is, if you do want to get involved, Ask BioPhorum is a very valuable tool that we would recommend you use. And that's not just for community practice. We recommend using that for all our work streams. It is an incredibly useful tool for benchmarking, for gaining quick information from the rest of the members, for all those who have the same expertise in your space. So it's very much a valuable tool we really do recommend using. So that is CPV again, in a nutshell. So that one's, with it being community practice, there's less going on. It's more about allowing the platform for our members to interact and discuss in the space and share what knowledge they have, what knowledge they are learning, what they're lacking, just to really understand from each other what everyone's doing and discuss any emerging trends as they come up. So again, if anyone is interested in taking part in this group, please do reach out to myself or through your account manager, and we can get you on board to the team. In the meantime, though, does anyone have any questions? I will assume silences, cool. No hands, no nothing. So we'll pass back to the wonderful Nicola.
Thank you so much, Sean. Thank you also to Olivia and to Hesam for those brilliant overviews. So I will just close then to share a little bit more on how to get involved. So depending if you are already involved in BioPhorum teams or not, this is a slide we put together to really describe how different the involvement might look for different individuals in BioPhorum, and we really can tailor it to the needs of your individual SMEs and to our member companies. So most importantly, it is about starting at your own pace. So whether you may be very new at the outset of your career, new into an organisation, or just new into BioPhorum, it's absolutely fine to join our teams just to listen in, join the workstream calls, observe what's going on. It's a really great way, particularly for new SMEs, to develop actually through the interaction with their peers across industry. You suddenly have this community outside of your normal peer group in your own organisation with tens of years of experience in their particular field, and your facilitator. So we've met two of the facilitators from the DS team today in Olivia and Sean, are here to support and can always have a little catch up one-to-one on how things are going and how things work in the work stream and how to get involved. But we do hope it will, for anyone getting involved in BioPhorum, expand beyond listening in. We do really invite you to bring your challenges. It works best when you are part of the community and coming along to those team meetings and presenting. Share case studies, stuff that you're experiencing right now, things that you have challenges with, and use the team to help you benchmark or validate what you're doing internally. We do hope as well for anyone participating in BioPhorum that you feel able to represent your organisation in influencing our programs, to be able to drive change where it matters most for you, and also hopefully share your knowledge to help influence the direction for our forum. And then in terms of contributing, there's so many ways to do this. So we've heard today already about the different publications and tools that the teams are working on and have a significant back catalogue in BioPhorum Drug Substance. This is just one way really to contribute. So by all means, we like to see our SMEs as parts of an authoring team, but also there's smaller ways in presenting case studies. The publications all obviously require review. We have a very healthy participation in Ask BioPhorum and our different surveys, so many different ways that you can contribute. And we hope as well for our SMEs that are engaged in our work streams to give you the opportunity to represent BioPhorum. So it's really quite often the case that as BioPhorum we can represent a cross-industry position and therefore a very strong position in influencing the industry, driving change, getting in front of regulators. This is a very powerful way that as a BioPhorum collaboration, we can make that difference. And we do hope that the final point on this slide around challenging the status quo, hopefully for those of you who are already involved in our BioPhorum teams, you do experience this winning team environment. The facilitators always talk about how much fun they have in their calls and at their face-to-face events, but we do try and very hard to create this safe space where it's very fun but respectful and we can be bold. It's step outside the day-to-day, outside of your organisation, think differently and challenge one another to do so as well. So just rounding up, a little summary there on the different ways to get involved. Thanks again to our presenters today. Please do follow up with one of us if you have any questions, you've heard anything that you would like to get involved with. But otherwise, I will close the session there. A few minutes back to everyone at a very busy time of year. Thank you so much, all of you, for your time, and we hope to see you again soon in BioPhorum. Thank you very much.